morning all. I am Dr. Dhondiba, Assistant Professor of Chemistry, Government First Grade College, Capital, Bangalore. Today I am going to discuss about 33 session of Biochemistry, which is Chemistry 8th, belong to Metabolism of 6th semester BSc. In this session, I am going to discuss about gluconeogenesis, gluconeogenesis, in which definition of gluconeogenesis, synthesis of glucose from lactate, fatty acid metabolism, activation of fatty acids, and role of carnitine. In this session, I have to discuss one by one. Now, first I am going to discuss about gluconeogenesis. Gluconeogenesis. What is mean by gluconeogenesis? The synthesis of glucose from non-carbohydrates. Non-carbohydrates, usually the carbohydrates contain glucose, no? But other than that is a compound contains glucose from carbohydrates, they are synthesized. Therefore, synthesis of glucose from non-carbohydrates compound is known as gluconeogenesis. Gluconeogenesis, which is a non-carbohydrate compound. Usually, in carbohydrates contain glucose already, but this will be a glucose can be obtained by synthesized from other than carbohydrates, called that is a gluconeogenesis. The precursor for gluconeogenesis are lactate, pyruvate, amino acids, and glycerol. From these are the reactants will be there. From this, the glucose molecule can be synthesized. Now, gluconeogenesis closely resembles the revealed reversed pathway of glycolysis. The gluconeogenesis closely resembles the reversed pathway of glycolysis. Although it is not a complete reversal of glycolysis, but partial it is a reversal. Reversal reactions of glycolysis are common for both glycolysis and gluconeogenesis, as they are reversible. Three reactions of glycolysis catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase are not reversible and require the involvement of four other enzymes, namely glucose 6-phosphatase, glucose fructose 1,6-diphosphatase, phosphate, pyruvate, then pyruvate carboxylase. This is the fructose 1,6-diphosphatase. Actually, ASE it should be there, phosphatase, pyruvate carboxylase, phosphoenol pyruvate carboxykinase. These are the enzymes participate in the conversion of the reactions. Now first I have to discuss one by one. Here lactate, how the lactate is converted into glucose here. Now lactate is obtained from that is lactic acid. Here lactate, this is the undergo a lactate dehydrogenase, and it is converted into pyruvate. Pyruvate will be formed. Now here just the hydroxy group is converted into that is ketone group by removal of that is undergo that is a dehydrogenation. Dehydrogenation, removal of hydrogen, and lactate dehydrogenation takes place and form that is a pyruvate. This pyruvate molecule undergo a pyruvate decarboxylation, means removal of carbon dioxide takes place using a ATP molecules, and that is a bicarbonate ion, and to convert into a that is oxaloacetate, and ADP and carbon dioxide. Carbon dioxide gas will be liberated and ADP is converted into ADP, adenosine diphosphate, and with liberation of that is a oxaloacetate. This oxaloacetate further undergo a phosphoenol pyruvate decarboxylase. In which addition of that is gyanosine triphosphate will takes place to this and gyanosine triphosphate is converted into gyanosine diphosphate with liberation of that is carbon dioxide, and this oxaloacetate is converted into phosphoenol pyruvate. Phosphoenol pyruvate. This phosphoenol pyruvate undergo a reversible reaction, that is a enolase. Enolase it takes place. That is a enolase form, that is conversion of that is alcoholic group is converted. That is enol form, that is a double bond alkene, that is unsaturated bond is converted into saturated. It is a function. Keto enol bond will be formed. That is actually here enol to that is the alcohol bond. It is formed. Therefore, in presence of the magnesium plus 2 as a cofactor, enolase will be formed, and this is the water molecule. Here, water molecule will be added, and to form that is here 2-phosphoglycerate. 2-phosphoglycerate will be a form, and this is the reversible reaction. The removal of that is water, it again form that is a phosphoenol pyruvate, and this 2-phosphoglycerate is further that is a phosphoglycerate mutase. It is undergo that is in presence of this enzyme, it convert 2-phosphoglycerate into 3-phosphoglycerate. 2-phosphoglycerate into 2-phosphoglycerate. It undergo that is just it is a mutase, means that is exchanging the group, that is the inter that is a rearrangement will be takes place. It is again a reversible reaction. Therefore, in this step, the 2-phosphoglycerate is converted into 3-phosphoglycerate by using a phosphoglycerate mutase as a enzyme. Then this 3-phosphoglycerate is further undergo a conversion to that is a 1,3-diphosphoglycerate using a phosphoglycerate kinase as an enzyme, using a ATP and to produce that is a 1,3-phosphoglycerate with liberation of ADP, conversion of ADP. Again, this is the reversible reaction. This 1,3-phosphoglycerate further it is converted into glyceraldehyde 3-phosphate by using a glycerol dehydrate 3-phosphate dehydrogenase. In this, the NADH is added, that is takes place added, and it converts into a this is the reduced form into oxidized form of the NAD plus phosphate, inorganic phosphate will be formed. Now here it converts, that is 1,3-diphosphoglycerate is converted into glyceraldehyde 3-phosphate by using a glycerol dehydro 3-phosphate dehydrogenase, and that is NADH is used to convert that is the NAD plus inorganic phosphate. Then this it is undergoing isomerization. Isomerization will be interconversion will takes place. Interconversion, just one position will be changes to other. Interconversion rearrangement will be takes place. Glyceraldehyde 3-phosphate is undergo interconversion to form that is a dihydroxyacetone phosphate. Dihydroxyacetone phosphate, and this again here too, this is the glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate is again condensed. Both it is combined with each other as a aldase as an enzyme to form that is a fructose 1,6-phosphate. Fructose 1,6-phosphate 1,6-diphosphate here. Fructose 1,6-diphosphate. It is not only phosphate. Fructose 1,6-diphosphate will be formed, and this fructose 1,6-diphosphate is further converted into fructose 6-phosphate by removing that is a phosphate molecule from the conversion of ADP to ATP using a magnesium plus 2 ion as a cofactor and phosphofructokinase as a enzymes. Then this fructose 6-phosphate is further converted into glucose 6-phosphate by using a isomerism. Just there is a functional group exchange, that is a glucose 6-phosphate isomerase in which the glucose 6-phosphate will be formed. And this is further the glucose 6-phosphatase plus magnesium plus 2 as a cofactor, and ADP as a, that is the adenosine diphosphate, is converted into ADP, and lastly it converted into a glucose. And this complete, that is a reaction, is takes place from conversion of, that is a lactate to glucose. How it is, several steps will be takes place. I have discussed details. Now coming to, that is a metabolism of fatty acids. What is mean by metabolism of fatty acids? In both animals and plants, the fats serve as a principal and most efficient source of stored energy. The animal and plants, they are the fats, the both animals and plants, the fats are serve as a principal and that is most efficient source of stored energy, because plants are stored their energy in the form of fat in seeds, the plant seeds, there no their energy will be stored in seeds, whereas animals the fats will be stored in, that is the adipose tissue, whatever may be. The absorbed fat is utilized to provide energy, and the excess of fat is transported to depot, that is transported depot of storage in the form of adipose tissue. Here simple, the absorbed fat is utilized to provide the energy, and the excess of the fat is transported or deposited as a depot in the form of, that is a adipose tissue in the animal. Then here the fat plus water, the lipase, it is further it is necessary required, then it is converted into glycerol, and fatty acids will be liberated. The glycerol enters the glycolytic pathway. Glycerol here, liberated glycerol is enter into glycolytic pathway, whereas fatty acids are transported by serum albumin to the sites of the oxidation, mainly the liver. The fatty acids are then oxidatively degraded by the pathway involving the removal of 2 carbon atoms at a time. Here, the glycerol enters glycolytic pathway, whereas fatty acids are transported by the serum albumin to the sites of oxidation, mainly the liver. Mainly the liver. The fatty acids are then oxidatively degraded by the pathway involving the removal of 2 carbon atoms at a time from the carboxyl end of the molecule, and this is known as a beta oxidation pathway. What is mean by beta oxidation pathway? Means it is takes place by removal of 2 carbon atom from a carboxyl end at a time, and this is called as known as beta oxidation pathway. This pathway is recognized as a principal method by which fatty acids are oxidized. The oxidation occurs in mitochondria in liver cells, as it contains high concentration of the enzymes necessary for fatty acid oxidation. Then coming to that is the activation of fatty acids. Fatty acids are activated by acyl coenzyme A synthetase, which attaches a coenzyme A molecule to the carboxyl group of the fatty acid. This enzyme occurs either in cytoplasm or in the other mitochondrial membrane and uses a molecule of ATP. The overall reaction is made irreversible by the subsequent hydrolysis of pyrophosphate to 2 molecules of the inorganic phosphate. Therefore, 2 equivalents of ATP are used up for this reaction. Therefore, here fatty acid react with coenzyme A thiol. It is undergo a reaction by using acyl coenzyme A synthetase, and ATP is used in here for activation, and ATP is converted into, that is AMP, adenosine monophosphate plus pyrophosphate. It forms with liberation of fatty acyl coenzyme A. Then coming that is the transport of fatty acyl coenzyme into mitochondria, or it is also called as a role of carnitine. Now here long chain fatty acyl coenzyme A is cannot cross the inner mitochondrial membrane. This is long chain fatty acyl coenzymes cannot cross the inner mitochondrial membrane into the matrix. Short and medium chain fatty acid acyl coenzyme molecules up to 10 carbon atoms can diffuse through. They have to be transported enzymatically across the inner mitochondrial membrane with the help of a substance called carnitine. It is the help of they are transported. Therefore, I say once again, read it. Long chain fatty acyl coenzymes cannot cross the inner mitochondrial membrane into matrix. Matrix is nothing but it is a short and medium chain fatty acyl coenzyme molecules up to 10 carbon atoms can diffuse through. Okay, matrix. But here, this is the long chain fatty acid, therefore they cannot be enter. They have to transport it enzymatically across the inner mitochondrial membrane with the help of substance called carnitine. Carnitine is help to transport the long chain fatty acyl coenzyme into mitochondrial membrane. Therefore, the carnitine serves as a transport agent in carrying the molecule across the membrane. Now here, this is carnitine react with the fatty acyl coenzyme. It is a carnitine acyl transferase will take place and it converted into fatty acyl carnitine plus coenzyme A thiol will be formed. So in this session, I have discussed about the gluconeogenesis and its definition, how to synthesize the glucose from lactase, that I have explained. Then fatty acid metabolism, in which activation of the fatty acids I have already explained. Then what is the role of that is carnitine, already I have explained in detailed manner. I hope you have understood. Thank you.